Hello friends, welcome to Gluconeogenesis part 2nd. In the part 1st, we were dealing with the substrates involved in gluconeogenesis. Now gluconeogenesis, as we know, it's the synthesis of glucose from non-carbohydrate substances, and this process is essential for the survival of living cells and living tissues because it ensures the normal blood glucose level. The major gluconeogenic tissue involved is liver as well as renal cortex, and the pathway is partly mitochondrial and partly cytosolic, and endoplasmic reticulum is also involved. Now the process is not a simple reversal of glycolysis, it involves much more enzymes, and we will see. So let's go. Now first of all, let's go through the key significance of gluconeogenesis. Now brain, cell system, erythrocytes, testes and kidney medulla, they are totally dependent on glucose for a continuous supply of energy. Usually, human brain requires around 120 grams per day of glucose. It consumes 120 grams per day of glucose, and the rest of the body needs 40 grams per day, which means out of total of 160 grams, brain alone utilizes uses 120 grams of glucose. And further, skeletal muscle also, especially during strenuous exercises, needs glucose because anaerobic glycolysis is what depends on what skeletal muscle depends. There is another scenario during starvation when body store of glycogen, the reserve store of glycogen will be depleted within 18 hours. And if the starvation continues, during prolonged starvation, fats will be utilized for a few days, and depending on the bulk of the body, protein will also be broken down after a few days. So this is what happens, or the substrates of gluconeogenesis. So this has been dealt in a separate module. You can go through it. Lactate amino acids, gluconeogenic amino acids, propionyl coenzyme A, glycerol. All these become substrates for gluconeogenesis. And before we actually see gluconeogenesis, we have to go through glycolysis. So this is the entire reactions of glycolysis. You can see glucose giving rise to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6 bis-phosphate, glyceraldehyde 3-phosphate, 1,3 bis-phosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and then pyruvate. So this is glycolysis. The challenge here is the three irreversible reactions. These three irreversible reactions here are highly hexagonic. You can see here the first one, that is catalyzed by hexokinase, glucose to glucose 6-phosphate. The energy release is negative 33.5 kilojoules per mole. This releases this much of energy. And the second one, which is catalyzed by phosphofructokinase first. This is fructose 6-phosphate to fructose 1,6 bis-phosphate. The energy release is negative 22.2 kilojoules of joules per mole, and that is also hexagonic. And the third one here, the third irreversible or the third key step of glycolysis is that catalyzed by pyruvate kinase, and this actually catalyzes the conversion of phosphoenol pyruvate to pyruvate. The energy release is negative 16.7 kilojoules per mole. Again, highly hexagonic. So totally 72 kilojoules are released. So bypassing these three steps was the challenge, and that's exactly what happens in gluconeogenesis. And it can never be as I said, it can never be a simple reversal of glycolysis. And also we have to look this TCA cycle also, just a reminder of the TCA cycle. You can see here the pyruvate is involved, which gets converted as coenzyme A and goes through all the cycle to give rise to oxalase state. Now oxalase state is the one which combines with coenzyme A to give citrates here. So this is just a reminder, two molecules here. One is pyruvate, another one is oxalase state. These two are the key molecules which we have to see. This is pyruvate. This is a 3-carbon compound. It's a keto acid, as you can see, double bond CO, C double bond O actually. Now pyruvate, and this is a 3-carbon compound, whereas oxalase state, it's a 4-carbon compound, again another keto acid. So these two compounds are very important, very key in the entire process of gluconeogenesis. Now, so as we saw in glycolysis, there were 3 steps which were catalyzed by hexokinase, phosphofructokinase first, and pyruvate kinase, which were irreversible and highly hexagonic. So what is the strategy adopted by cells? The strategy is gluconeogenesis, as we know, and the pyruvate molecule cannot be converted back to phosphoenol pyruvate. So what here is followed is, first of all, it will be converted to oxaloacetate, and that is done with the help of the enzyme pyruvate carboxylase. And the pyruvate carboxylase, we will see the reaction in detail later. Pyruvate carboxylase, it adds a carbon it adds a carbon to pyruvate to give rise to the 4-carbon oxaloacetate. And the next step involves phosphoenol pyruvate carboxykinase. Obviously, 1 carbon is lost to give rise to phosphoenol pyruvate, because phosphoenol pyruvate is a 3-carbon compound. So that also we will see in detail. And the next key step, for that of phosphofructokinase first, that is surpassed with the help of fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase. So that is a hydrolytic enzyme, obviously. And another one here is glucose 6-phosphatase, another hydrolytic enzyme. And this one surpasses the first step of glycolysis, namely glucose giving rise to glucose 6-phosphate. That is reversed to give back glucose from glucose 6-phosphate. So these are the 3 reactions which we have to bypass. First one, the second one, and the third one. And first bypass reaction is conversion of pyruvate to phosphoenol pyruvate. This actually takes place in 2 steps. So first bypass, step 1, which is actually formation of oxaloacetate from pyruvate. And pyruvate, we know it's the end product of glycolysis, which is released in cytoplasm. Obviously, glycolysis takes place in cytoplasm. And pyruvate but can cross mitochondrial membrane to reach mitochondrial matrix. So that's where the whole reaction takes place. And HCO3, it is actually formed from carbon dioxide and water with the help of carbonic anhydrase. And carbonic acid formed instantaneously splits to HCO3 minus. So that is available here. And obviously, the original reaction of pyruvate adding carbon dioxide, this is an endergonic reaction, and so cannot move forward. So as happens in metabolic reactions, a high energy ATP is hydrolyzed. A high energy molecule, here it's an ATP molecule, is hydrolyzed, which actually it's an hexagonic reaction. As you know, hydrolysis of ATP is hexagonic. And the combined delta G becomes negative and hexagonic, and so the reaction can move forward. And this is done with the help of the enzyme pyruvate carboxylase. This is an enzyme having 4 identical subunits to give oxaloacetate. When we go further into the mechanism of the reaction, what happens actually here is the initial step. In the initial step, ATP combines with bicarbonate so as to give carboxyphosphate, which releases ADP. So this is what happens first. And this carboxyphosphate, this phosphorylated carbon dioxide can attach onto biotin enzyme. So this is what happens first. Biotin enzyme to give an intermediate complex, which is carboxy biotin enzyme intermediate. So this is what happens first. And the bond here in this molecule is very unstable, and it can give out an energy of negative 20 kilojoules per mole on breakage. So this is what drives the reaction forward, being hexagonic. This being hexagonic. So I mean, this is a kind of activated intermediate. This one is an activated intermediate. Since this energy released can be used for the reaction to proceed further. And this kind of strategy is the strategy of activated molecules can be seen in usually in metabolic process in biochemical reactions. One of them is, as you may remember, in glycogenesis, UDPG is the activated form of glucose there. UDPG is formed by activating glucose with the help of UTP. Here it is ATP. Now eventually what happens is that in this intermediate reacts with pyruvate, and so as to carboxylate it, and the product is oxaloacetate. So that is what happened, like this here, and the oxaloacetate formed here is the released oxaloacetate is released inside mitochondrial matrix now. So this has to be taken to cytosol, and mitochondrial matrix is impermeable, or oxaloacetate cannot cross through the mitochondrial membrane, but malate can go through the mitochondrial membrane, because the malate transporter protein is available in the mitochondrial membrane. So malate dehydrogenase, which is present in both in mitochondrial matrix as well as in cytosol, that is the enzyme involved in the process. Oxaloacetate is first converted to malate, and malate traverses the membrane so as to reach cytosol, and it gets converted to oxaloacetate along with the production of NADH. So what happens is that at one side oxaloacetate is reduced to malate, whereas this NADH is oxidized, and on the other side, on the cytosolic side, malate gets oxidized to oxaloacetate, at the same time reducing NAD plus to NADH. So eventually what happens is that oxaloacetate gets transported to cytosol, and so oxaloacetate is present now in cytosol. So step 2 is here two things are achieved. First one is decarboxylation of oxaloacetate here, decarboxylation of oxaloacetate, and second one is phosphorylation of phosphorylation also can occur with the help of GTP. And obviously here also the strategy is the same, because GTP is hydrolyzed so as to make the entire reaction hexagonic and to make the reaction move forward. And decarboxylation alone is obviously endergonic. This process is endergonic, so that's why it is coupled with the breakage of high energy bond of GTP, so that phosphoenol pyruvate is formed, so thus completing the first bypass reaction. So so this is what happens. We move on to so this is the effective reaction. Now pyruvate gets converted to phosphoenol pyruvate. So we can go through it, just pause it. And the second bypass reaction. So this is a conversion of fructose 1,6 bisphosphate to fructose 6-phosphate. This is a hydrolytic reaction involving an enzyme belonging to hydrolysis, namely fructose 1,6 bisphosphatase, which here fructose 1,6 bisphosphate, it is hydrolyzed, give rise to fructose 6-phosphate. And the next reaction, the final third bypass reaction, this also involves hydrolysis, that is conversion of glucose 6-phosphate to glucose. So glucose 6-phosphate gets converted to finally to glucose with the help of the enzyme glucose 6-phosphatase. This also is an enzyme belonging to hydrolysis. But here glucose 6-phosphate is present in liver, in kidney. It is not present in much of the other tissues. In cells, the enzyme occur membrane bound in the endoplasmic reticulum. So this is endoplasmic reticulum, the lumen, cytosolic side. It is membrane bound here. And it is notably absent in muscle, adipose tissue, brain. And this is why mainly liver can replenish or restore blood sugar level through gluconeogenesis. So that's happened. This is what happens. Glucose 6-phosphate converted to glucose. So glucose is available now. Glucose 6-phosphate is the enzyme. So it goes through and reaches the cytosolic side, so that it can be released to blood from hepatocytes. To sum up, 2 molecules of pyruvate, because pyruvate is a 3-carbon compound, give rise to glucose, which is a 6-carbon compound, and the rest of the molecules involved are, we have discussed them. And the energy requirement, the stoichiometry of gluconeogenesis is a total of 4 ATP to GTP. So this process is, gluconeogenesis is really costly. It involves 6 ATP or expenditure of 6 ATP as well as loss of 2 NADH molecules also. Okay, thank you.